Dear students, today's topic is Hardy-Weinberg Principle. This is very important, and one question is always asked from this topic. Hardy-Weinberg Principle. It was given by, as the name suggests, G.H. Hardy and W. Weinberg, Wilhelm Weinberg, in 1908, given by G.H. Hardy and Wilhelm Weinberg in 1908. Principle. The statement of principle, it states that the allelic frequencies in a non-evolving population are stable and remains constant generation after generation. It means in a population, the non-evolving population, the allele frequencies remains all stable and constant generation after generation. That is, the sum of all allele frequencies is 1. So this is the statement. This is the statement, and sometimes it is asked that write the statement of Hardy-Weinberg principle. The mathematical expression. Mathematical expression. P plus Q is equal to 1. If it is elaborated or expanded, then P square plus 2PQ plus Q square is equal to 1, or P plus Q is equal to 1. These are the expression. This is the main expression, and it is only the expanded form of this, and then you may write like this. What is P? P square, it indicates frequency of homozygous dominant trait, and then Q square, it shows the frequency of homozygous recessive trait, and the 2PQ, it indicates the frequency of heterozygous individual in a non-overlapping, non-evolving populations. It is very important. By taking one example or by taking one questions, I can explain it more. Question, that is, in a sampled population, the population, the frequency of dominant trait is 64%. Calculate number 1, frequency of homozygous recessive trait in percent. Frequency of heterozygous individual. According to question, p square is given 64%. The p square is equal to 64 by 100, then p is equal to 64 by 100 is equal to 8 by 10 is equal to 0.8. Now, as per the, according to Hardy-Weinberg Principle, P plus Q is equal to 1, and you have found out, putting the value of equation 1 in equation 2, so 0.8 plus Q is equal to 1. Q is equal to 1 minus 0.8 equals to 0.2. So then frequency of homozygous recessive trait is Q into Q is equal to 0.2 into 0.2 equals to 0.4 equals to 0.04. 0.04 equals to 4%. Number 2, frequency of heterozygous individual, frequency of heterozygous individual is 2 into P into Q. 2 into P is 0.8 into Q is 0.2, then the total is 32. 0.32 is equal to 32%. Now Hardy-Weinberg principle or equilibrium. Hardy-Weinberg principle or equilibrium is affected by five factors, or you may say factors affecting Hardy-Weinberg principle, equilibrium. Number one, mutation. It is a sudden change, and it may cause deletion or addition of gene or allele of a particular certain trait, which may affect Hardy-Weinberg principle. And the number two, gene flow or gene migration. And then number three is a recombination. Number four, natural selection. And then number five is genetic drift. Now question is, what is genetic drift? Genetic drift, elimination of gene, elimination of gene or any change in gene of a population due to migration or death is called genetic drift. Means if any gene is either eliminated, removed from a gene pool of a population, or gene may be altered, and it may happen due to migration or death. This is called genetic drift. Genetic drift cause number one, elimination of certain allele. Number two, preponderance of another allele. Means one gene or one allele can be removed and another allele of a certain trait can be increased. And the number three, survival population has a small gene pool, because due to elimination or due to death or due to migration, some individuals, some species move from that place and cause a small gene pool. This increases inbreeding, increases next heterozygosity, decreases and homozygosity increases. Favor allele is present. No, as if the population will be small, if the population size is small and certain genes are eliminated, then what happens? There is less chances of outbreeding, and the animals or the individuals will be compelled to do inbreeding, and that will increase homozygosity and will decrease heterozygosity. Now the next one is founder effect. This is also very important. Founder effect, genetic drift, gene migration. A genetic drift in human population where where a population where a population where a new population is settled, which has different gene frequencies, entirely new populations. This is called, and the original drifted population, the original drifted population is called founder, and this effect is known as founder effect. It can be easily explained by this example. It can be easily explained. Suppose this is original population. Of species A, B, B, C, C, D, and D. Due to some reason, this portion is separated like this. This portion has B, C, A, D, and this portion has A because C and B dies due to some. Or here is only C, two populations. The remaining two populations are died or migrated to this place. Migrated to this place. This is migrated. This is migrated. Now, this population of this place is called founder. It is small in size, less population, and this is called founder populations. And now the next one is bottleneck effect. Bottleneck effect. This is also called right effect, and this is also called genetic bottleneck. Genetic bottleneck. Due to genetic drift, new population is established, which has poorer gene pool than original population, and less variability, reduced variability and adaptability, leading to extinction of species. This effect is called bottleneck effect. जेनेटिक ड्रिफ्ट के जरिए जो न्यू पॉपुलेशन बना, तो उस न्यू पॉपुलेशन में जीन पूल वहाँ पर भी है, लेकिन जो ओरिजिनल जीन पूल था उससे पुअर है वो। दूसरी बात, उसमें वेरिएबिलिटी भी कम भी पाई जाती है, एडेप्टिव नेचर भी, चूँकि न्यू पॉपुलेशन है इसलिए एडेप्टिबिलिटी भी कम पाई जाती है, जिससे कि स्पेसीज के एक्सटिंक्ट होने का चांस बढ़ जाता है। इसी को बॉटलनेक इफेक्ट कहते हैं। थैंक यू। इट विल रिमेन कंटिन्यू।